Hello everyone, welcome back. So let us continue with our series. So हम थे हमारे सेक्शन 3 में और लास्ट टाइम हमने पढ़ा था अबाउट वायरसेस एंड देयर क्लासिफिकेशन एंड टुडे लेट अस स्टार्ट विद बैक्टीरिया, आर्किया एंड देयर ब्रॉड क्लासिफिकेशन. यूकैरियोटिक माइक्रोब्स जो है वो ज़ूलॉजी और माइक्रोबायोलॉजी दोनों में कॉमन है और आज हम लोग पढ़ेंगे अबाउट बैक्टीरिया एंड आर्किया. बैक्टीरिया इज अ वेरी बिग चैप्टर. उसमें इन डिटेल्स पढ़ना पड़ता है बैक्टीरियल सेल्स के बारे में. वो थोड़ा बाद में देख लेंगे हम लोग. फिलहाल आज हम देखेंगे क्लासिफिकेशन, बैक्टीरियल क्लासिफिकेशंस एंड आर्कियल क्लासिफिकेशंस. बैक्टीरियल क्लासिफिकेशन. बैक्टीरियल क्लासिफिकेशन में जाने से पहले लेट अस नो अबाउट बर्जीज मैनुअल ऑफ सिस्टमैटिक बैक्टीरियोलॉजी. ये लिखा था डेविड बर्जी ने और उन्होंने प्रॉपर्ली क्लासिफाई किए थे प्रोकैरियोट्स को डिपेंडिंग अपॉन देयर कैरेक्टर्स, डिपेंडिंग अपॉन देयर सर्वाइवल एक्सेट्रा. तो बस ये एक वन लाइनर है. The first classification is based on metagenomics. Metagenomics मतलब studying the structure and function of nucleotide sequences, DNA sequences, इन सब की पढ़ाई को मेटाजीनोमिक्स, दिस स्टडी इज नोन एज मेटाजीनोमिक्स. और मेटाजीनोमिक्स के अंडर फॉर बैक्टीरियल क्लासिफिकेशन वी स्टडी 16S RNA सीक्वेंस क्लासिफिकेशन ऑफ बैक्टीरिया. और इस क्लासिफिकेशन में बैक्टीरिया को डिवाइड किए थे डिपेंडिंग अपॉन देयर नीश इकोलॉजिकल environment. Example, rainwater bacteria, well water bacteria, or bacteria is found in mountains, hot streams, etc. So याद रखना है यहाँ पे 16S RNA sequence was used in metagenomics for classification of bacteria. Next classification was metabolism. मेटाबॉलिज्म बैक्टीरिया में दो टाइप का हो सकता है. नॉर्मल सेल्यूलर मेटाबॉलिज्म विच इज एरोबिक, दैट इज ऑक्सीजन इज प्रेजेंट, और fermentation that is carried out under an aerobic condition. Oxygen is not present. So इस बेसिस में हमने बैक्टीरिया को क्लासिफाई, वी कैन क्लासिफाई बैक्टीरिया into 5 different classes, and the sixth one we'll see that later. So first one is called as obligate aerobes. Obligate मतलब strict, aerobes मतलब oxygen, so they strictly need oxygen for their survival. Without oxygen they won't survive. अभी ये जो ट्यूब है उसमें है thioglycolate medium. This is a medium that is used to grow bacteria. So you can always see oxygen कहाँ present होगा, यहाँ air में. So here at the surface some amount of oxygen will be present, but over here at the end of the tube there won't be any oxygen. Oxygen यहाँ तक पहुँचेगा भी नहीं. So we can see that this is a aerobic condition, and here it will be a anaerobic condition. So in case of obligate aerobes, most of the bacteria is seen at the top of the medium. Second is facultative aerobes. Facultative aerobes are the ones which can survive in the presence of oxygen or in the absence of oxygen, but they प्रिफर ऑक्सीजन, मतलब वो मोस्टली, एक मिनट, ओके दिस डायग्राम इज रॉन्ग, आई थिंक ये इंटरचेंज हो गया, उन्होंने इंटरचेंज कर दिया. दिस डायग्राम इज ऑफ obligate aerobes and this is for facultative anaerobes. So facultative anaerobes में क्या होगा? Organisms will be present mostly on the top of the medium because they prefer oxygen even though they can grow in the absence of oxygen. So नीचे भी थोड़े microorganisms present होंगे but mostly ऊपर होंगे. Third one is obligate aerobes. Obligate anaerobes. The strict anaerobes, so oxygen is toxic from them, oxygen is poisonous, so they cannot grow on the surface. So यहाँ पे organisms नहीं है, so सारे organisms यहाँ पे नीचे पाए जाएंगे जहाँ बिल्कुल oxygen नहीं है. Please note that this diagram has been interchanged. Next is aerotolerant anaerobes. Aerotolerant मतलब they can tolerate, मतलब ऑक्सीजन अगर है अवेलेबल है इन द मीडियम दे कैन टॉलरेट. सो इवन इफ लाइक ऑक्सीजन नहीं है तो उनके लिए बेस्ट है, दे विल ग्रो इन द एब्सेंस ऑफ ऑक्सीजन, बट अगर है तो भी टॉलरेट हो जाएगा. सो दे कैन इग्नोर ऑक्सीजन, मतलब एक ऑक्सीजन है तो इग्नोर कर देंगे, अपना खुद का मेटाबॉलिज्म करते रहेंगे. दिस इज द एरोटॉलरेंट एनारोब्स. देन कम्स माइक्रो एरोफाइल्स. माइक्रो एरोफाइल्स मतलब they require micro मतलब बहुत कम, so they require very less amount of oxygen. कितना? Only 2 to 10%. But less amount of oxygen, यहाँ तो बिल्कुल ऑक्सीजन नहीं है, यहाँ पे सबसे ज्यादा ऑक्सीजन है. Less amount of oxygen is present in the middle. So microaerophiles will be mostly found over here in the middle. So this was the 5 classes. Sixth one extra जो है, it is capnophilic bacteria which grows in the presence of carbon dioxide. This is also a similar chart जिसमें obligate anaerobes, facultative anaerobes, aerotolerant वगैरह डिस्क्राइब किए हैं. बस इसमें एक्स्ट्रा जो है they are the examples. Examples आ सकते हैं, तो I have listed some of the important examples which has high probability of coming. So थोड़े से तो examples याद रखने पड़ेंगे, जैसे obligate में Clostridium tetani और Clostridium botulinum. Then facultative मतलब प्रेजेंस और एब्सेंस दोनों में पाए जाएंगे, वो हम इजीली पाए जाते हैं, वो ई कोलाई एंड स्टेफाइलोकोकस ऑरियस. एरोटॉलरेंट इज लैक्टोबैसिलस एंड ई फेकालिस. एंड ऑब्लिगेट एंड एरोब्स जिसका ऑक्सीजन इज प्रेजेंस इज नेसेसरी, ऑक्सीजन प्रेजेंस नेसेसरी है तो वो कौन सा मेटाबॉलिज्म शो करेगा? इट विल बी ऑलवेज सेलुलर रेस्पिरेशन. एंड इफ देयर इज नो सेलुलर रेस्पिरेशन मीन्स इट इज इन द absence of oxygen, so they are anaerobes. Then microaerophilic example is H. pylori which causes ulcers, and Campylobacter. And capnophilic which require increase in the amount of carbon dioxide and decrease in the amount of oxygen. Example is influenza and gonorrhea. Third type of classification is based on nutrition, like how the bacteria obtain nutrition, and it can be, but it is further classified based on the source of energy. Energy कहाँ से ले रहे हैं? If it is light source or any other chemical source like that. So based on energy, source of energy, based on electron donor, donor कहाँ से मिल रहा है, और based on the carbon source, if it is organic source, inorganic source. These are the 3 broad classifications for nutritional diet. So under source of energy, it can be prototrophs which come under which grow under light, that is they require light as a source of energy. Then they contain various pigments. There are a number of pigments which have different functions, we will see them later. Then chemotrophs which require chemicals and they undergo chemical reactions such as oxidation. They undergo chemical oxidation. अभी इसमें भी they can undergo chemical oxidation and under organic or inorganic. ऑर्गेनिक मॉलिक्यूल्स के साथ. So organic molecules such as glucose, inorganic such as ammonia, H2S, Fe, etc. So इसके अंदर जो ग्लूकोज इजीली मेटाबोलाइज करता है केमिकल ऑक्सीडेशन से, एग्जांपल्स विल बी E. coli. And here inorganic source mostly will be nitrifying bacteria, denitrifying bacteria. We will study this. इससे भी questions आते हैं, ये दूसरे section का एक topic है. So उसके examples पढ़ेंगे हम लोग. बस अभी nitrifying bacteria याद रखना है ताकि उसके examples बाद में याद आए. Then hypotrophs. Hypotrophs मतलब they are parasitic. They depend on other. They suck nutrition from other microorganisms such as viruses and rickets. इनको hypotrophic organisms बोलते हैं. Next, based on the source of electron donor. So electron donor लिथोट्रोफिक हो सकता है या फिर ऑर्गेनोट्रोफिक. लिथोट्रोफिक मतलब इनऑर्गेनिक, और इनऑर्गेनिक में आएगा फिर अमोनिया, H2S, Fe, H2, और ऑर्गेनिक में ग्लूकोज एक्सेट्रा. The other organotrophs. Based on the carbon source. So the carbon that is required for the metabolism, they can obtain it through autotrophic. मतलब जो खुद से अपना खाना बनाते हो, they prepare food by themselves, autotrophs. So they take the atmospheric carbon dioxide. So the carbon source over here is carbon dioxide. Heterotrophs which depend on others. So the carbon source will be methane from others. And paratrophs, it is parasitic, and they taking the ready-made amino acid from other microorganisms. And examples include viruses, rickets, etc. So prototrophs under for them the source of energy is light, and they convert light into food with the help of certain pigments. And the most common pigment that we all know about is chlorophyll. But और भी pigments हैं, जैसे एक pigment है क्लोरोबियम, chlorophyll. Then there is bacterioviridin. Bacterioviridin is a pigment that is present in green sulfur bacteria. बस याद रखना है, bacterioviridin is present in green sulfur bacteria. Then there is bacteriochlorophyll that is present in purple sulfur bacteria or purple non-sulfur bacteria. Then there is bacteriopurpurin. Bacteriopurpurin is a pigment which is red in color. It is red in color and it reduces highly oxidized compounds. It is mainly responsible for reducing highly oxidized compounds by absorption of light. So this is responsible for absorption of light and reduction of oxidized compounds. कुछ नहीं, बस इन पिगमेंट्स का नाम याद रखना है और कहाँ पे प्रेजेंस है और उनके फंक्शंस क्या है. क्वेश्चंस आएंगे तो इन्हीं से आएंगे. ये कुछ नहीं है, ये एक चार्ट है. कुछ नहीं है मतलब इससे क्वेश्चन आए इसलिए रखा है. सो एक चार्ट है जिससे क्वेश्चंस पूछते हैं कि प्रोटोलिथोट्रोफ्स मतलब प्रोटो मतलब लाइट, is the source of energy. Litho, inorganic electron donor, or carbon source is carbon dioxide, like organic carbon source. So like that. Then proto organotrope, proto matlab light, organotrope matlab organic source of electron donor and carbon source. Heterotroph, proto heterotroph is light and organic source. Then chemo is chemical source of energy, like that. ये भी सेम चार्ट है, जस्ट इसमें थोड़े एग्जांपल्स है कि फोटोलिथोट्रोफ्स में पर्पल सल्फर बैक्टीरिया आएगा और ग्रीन सल्फर बैक्टीरिया आएगा. देन फोटो ऑर्गेनोट्रोफ्स में पर्पल नॉन सल्फर बैक्टीरिया एंड फोटो ऑटोट्रोफ्स साइनोबैक्टीरिया. फोटो हेट्रोट्रोफ्स में पर्पल नॉन सल्फर बैक्टीरिया एंड देन देयर इज आर्किया. Then the next category that is there, a small category of microorganism is mixotrophs. That is, they are mixed. That is, they can utilize both inorganic plus organic source of electron donor. Then another important point is Rhodospalium rubium. It is a bacteria which acts as both phototroph, that is light as a source of energy, and chemotroph, the chemical से भी energy source use करती है. Then we'll be knowing about oxygenic photosynthesis and anoxygenic photosynthesis. So oxygenic photosynthesis is the one wherein oxygen is reduced, and oxygen wherein oxygen is not reduced. Oxygenic photosynthesis is a non-cyclic photosynthetic reaction. And why oxygen is produced? Because here water acts as a electron donor, inorganic source of electron donor. But in an oxygenic photosynthesis, here H2 is not the electron donor. So some other organic source will be there as the donor of the electron. Examples of bacteria undergoing anoxygenic photosynthesis is green sulfur bacteria, purple sulfur bacteria, purple non-sulfur bacteria, and chlorobium. And here, like in oxygenic, the first bacteria to show respiration in bacteria, it is cyanobacteria. Next are some examples of chemoorganotrophs, chemolithotrophs, and phototrophs. These are the ones which have like at least higher chances of coming as the options in your question. So you should get familiar to their names. You can remember, like in case of chemolithotrophs, mostly it is methane producing bacteria like Sarcina, Methanococcus, or Methanobacterium. And in case of chemoorganotrophs, mostly it is Clostridium, desulfovibrio, desulfobacteria, or desulfomuricin. So mostly sulfur producing, it will be chemoorganotrophs. And phototrophs are Chromatium, Thiocapa, Chlorobium, and Heterotrichospira. So chroma color or chlorophyll containing is they are mostly phototrophs. Next classification is based on lactose fermentation. Lactose fermentation is mostly checked using McConkey agar. Other media is also used, but most commonly used is McConkey agar. And उसका presence और positive response हमें दिखता है as pink colonies. So here this is lactose fermenting colonies, and colorless रह गया like this. This is the media color actually. It is non-fermenting, non-lactose fermenting colonies. So lactose fermenter, so इसमें classify होते हैं as lactose fermenters, late or delayed lactose fermenters, and non-lactose fermenters. Lactose fermenters में E. coli, Klebsiella, and Enterobacter. And late or delayed lactose fermenters में Shigella, Salmonella, Shigella, and Paracolons. Non-lactose fermenters में Salmonella and Shigella दोनों आते हैं. Then non-lactose fermenters can be further classified as non-motile, non-H2S producers. मतलब H2S नहीं प्रोड्यूस करते और मूव भी नहीं करते, non-motile है. Example is Shigella. And motile H2S producer is Proteus and Salmonella. So Shigella क्या है? It is non-motile and non-H2S. And Salmonella is motile H2S producer. So lactose fermentation, positive matlab lactose fermenters and negative is non-lactose fermenters. Then further it is motile or non-motile. इसमें भी motile and non-motile है, and H2S producing or H2S not producing. इसमें just examples है थोड़े, वो याद रख लेना. Next classification is based on the catalase activity. Catalase is an enzyme that is responsible for catalysis of H2O2 to H2O plus O2. And this catalase test helps us to detect the presence of the catalase enzyme. So, ये catalase enzyme इस reaction को catalyze करता है. So, it can be catalase positive, catalase present है, so catalase negative, that is catalase absent, and catalase positive. So, catalase negative is Streptococcus and catalase positive is Staphylococcus. Then catalase negative is further classified as alpha, which undergoes partial hemolysis, or beta, which when it goes complete hemolysis, or gamma, wherein there is no hemolysis. और इसके examples यहाँ नीचे है, pneumonia, E. coli, then beta K, streptomyces pyogenes, and no hemolysis gamma is epidermidis. Then under catalase positive, it is coagulase negative or coagulase positive. Coagulase positive is हमारे Staphylococcus aureus. Next classification is based on the morphology. ये बस देख लेना, माइक्रोबायोलॉजी में पढ़ा होगा. एग्जाम्पल्स नोट करने हैं तो इनके करने हैं, जैसे क्लब रॉड में कॉर्नियोबैक्टीरियेसी, हेलिकल में हेलिकोबैक्टीरियो पाइलोरी, and Vibrio for comma shape. Okay, these are some examples. It contains some examples which can be learned, like Vibrio bacteria, comma shaped bacteria, helical bacteria, H. pylori. Then spiral is spirochetes and Treponema palladium. इससे क्वेश्चंस आए, so एग्जांपल से क्वेश्चन आते हैं इनमें से. So Coscre is Borrelia burgdorffi. Then club rod is Corneobacteriaceae. Then pleomorphic bacteria. Pleomorphic bacteria are the one which can change their shape. According to the environment, they can change their shape. They do not have a definite shape. Then actinomycetes, they are referred to as fungi-like bacteria because they appear filamentous. ऐसे लाइन में अरेंज होंगे जैसे फिलामेंट्स अरेंज होते हैं फंगाई के. सो दे आर कॉल्ड एज एक्टिनोमाइसेट्स. Next is mycoplasma. Mycoplasma is like the smallest way a microorganism which do not have a cell wall, and it is pleomorphic because it can change its shape. Pleomorphic मतलब shape change करना. Then it is like it does not have a cell wall, but it has a plasma membrane and membrane, and it is mycoplasma is more susceptible to osmotic shock or any detergent treatment or any toxic treatment, chemicals, etc. Rickets and chlamydia, these are also closely related to viruses, and these are obligate parasites. And let us stop for today. और भी classifications हैं, वो हम लोग नेक्स्ट लेक्चर में देख लेंगे. Next classification is based on temperature. This is an important classification. And depending upon the temperature in which the bacteria grows or where it is found, it is divided into the following. So first आते हैं हमारे साइक्रोफाइल्स. These are found at a temperature of 4 degrees Celsius. मतलब normal जो temperature for their growth is 0 degrees to 20 degrees. मतलब 0 to 15 consider कर सकते हैं, 20 consider कर सकते हैं. Then mesophiles जो ऑप्टिमम टेम्परेचर हमारे टेम्परेचर पे रूम टेम्परेचर पे ग्रो करते हैं, इट इज फ्रॉम 10 to 50 degrees approximately. Next is thermophilic, which is at a higher side, 40 degrees to 70 degrees. Hypothermophiles at a very high temperature, these are 70 to 100 और उससे भी ज्यादा, जो उनको भी हाइपोथर्मोफाइल्स करते हैं. स्पेसिफिक एग्जांपल इज पायरोडिक्टम ब्रोकी. इट ग्रोज एट द ऑप्टिमम टेम्परेचर ऑफ 106 डिग्री सेल्सियस. इनके एग्जांपल्स याद रखने पड़ेंगे. साइक्रोफाइल में फ्लैवोबैक्टीरियम, मेसोफाइल रूम टेम्परेचर पे ई कोलाई. Thermophilic is Bacillus stereothermophilus, and hypothermophiles is Thermococcus cever. Here there are some examples noted down. Examples याद रखना जरूरी है. साइक्रोफाइल्स में मोस्टली पोलेरोमोनास, सूडोमोनास, क्लैमिडोमोनास. सो लाइक रिमेंबर कि मोनास जिससे मोस्टली वो है साइक्रोफाइल्स में आते हैं. पोलेरोमोनास, सूडोमोनास, क्लैमिडोमोनास, साइक्रोमोनास एंड मिथेनोजेनियम. देन फैकल्टेटिव साइक्रोफाइल्स, मतलब साइक्रोफाइल्स जो है they are divided into two types, two true psychrophiles and facultative psychrophiles. True psychrophiles मतलब वो properly -10 to -15 डिग्री टेम्परेचर पे ग्रो करेंगे, एंड फैकल्टेटिव मतलब 0 to 35 टेम्परेचर तक जाएगा उनका. So facultative में हमारे there are certain fungi, food spoiling fungi, then pneumonia bacteria. Mesophiles, which grow on human temperatures, is human pathogens E. coli. Thermophiles, which is greater than 55, is certain algae and fungi, and Styrothermophilus and Globobacilli. Then hypothermophiles. Hypothermophiles are the ones which go beyond 90 degrees, beyond 100 degrees, and these are found in hot vents, sea floor, hot areas. Like example, Thermus aquaticus. And इसमें सारे जो मोस्टली हैं वो है पायरोकोकस, पायरोडिक्टम, थर्मोकोकस, पायरोलोबस. सो कोकस, डिक्टम एंड लोबस वो आते हैं हाइपोथर्मोफाइल्स में. Now it is interesting to note that even though we are studying a common group of microorganisms like that is bacteria, and they have similar cell structure, similar cell wall, and like they are common group, but still some of them grow under very low temperatures of -degree Celsius, while some of them find the optimum temperatures of 100. So why is there a difference in their adaptations? So first, let's see about psychrophiles. So psychrophiles grow at a minimum temperature, low temperature of 0 to 20 °C, और इसमें होता है two psychrophiles, and then there is facultative psychrophiles. So what is the difference? Psychrophiles, they have a cell membrane. उनके cell membrane have high level of unsaturated fatty acids. So cell membrane is made up of fatty acids, saturated or unsaturated fatty acids. So psychrophiles में high level होता है unsaturated fatty acids का. इससे क्या होता है कि they remain solid. The fatty unsaturated fatty acid, they remain solid at low temperatures. So they are solidified at low temperature or in cold conditions. And if the temperature increases then 20 °C, then the cell contents, मतलब वो melt होते, cell contents will melt because temperature is greater, and it will leak. This is in case of saturated fatty acids, which are higher in amount in psychrophiles. Also, they have a higher degree of alpha helix flexibility. And how will this affect the temperature? We will see this in the biochemistry section, because there is a detailed section for this. Then they have higher amount of polar and less hydrophobic amino acids. So polar amino acids and less hydrophobic amino acids. So these are the conditions or adaptations which help a psychrophile to live at very low temperatures. Then that was the case in psychrophiles. Now what is the case in case of hyperthermophilic bacteria, which grow under temperatures of 90 to 120 °C. So mostly they will have heat stable enzymes which will function even at very high temperatures. Then proteins, they will have heat stable enzymes and proteins which will function. Then they will have, since psychrophiles have high amount of unsaturated fatty acids, these will have more amount of saturated fatty acid. इससे क्या होता है? सैचुरेटेड फैटी एसिड्स, ये अगर बायोकेमिस्ट्री में पढ़ा हो तो सैचुरेटेड फैटी एसिड्स है वो higher melting point as compared to unsaturated fatty acids. This comes under the biochemistry of lipids. So this is in case of hypothermophiles, and they are also called as extremophiles. Extremophiles matlab they live under extreme conditions. It can be psychrophiles or hypothermophiles also. Also, one thing to note under psychrophiles is that they psychrophiles में होता है cryoproteins. Just like hypothermophilic bacteria contain heat stable proteins, here that is cryoproteins, which help in the adaptation of psychrophilic microbacteria at very low temperatures. Next is classification of bacteria based on the pH requirement. So pH, if it is 1 to 5, it is acidophile, that is acidic pH. 5.5 to 8, neutral pH. More than 8 to 11, it is alkalophiles. And then there is extreme alkalophiles, which have a pH greater than 10. Examples are important in this. Some examples are discussed over here. So in acidophiles, we see there is fungi, algae, cynidium, then Picrophilus, Lactobacilli, Salpolobus, etc. and Pyroplasma. Pyroplasma पे एक ईयर क्वेश्चन आया था. In neutrophiles, there is bacteria and protozoa. Alkalophiles like Ficales, then Nitrobacterium, Vibrio cholera, Agrobacterium. Extreme alkalophiles which grow under pH greater than 10, about 14-15, is Bacillus alkalophilus. Now let us see the adaptations for this, which causes pH fluctuations in these bacteria. So in case of neutrophils, which grow at an optimum pH of 5 to 7, so their plasma membrane is impermeable to protons, and there is H plus antipode, which is responsible for the exchange of potassium ions. Then in case of alkalophiles, which grow at a pH greater than 11, they exchange sodium ions for the in exchange for hydrogen ions, and there is an internal buffer which still maintains the uh, inner bacteria even under high pH conditions. In case of acidophiles, they contain a special protein which is called as a neoprotein. Neoprotein is special because a high acidic tolerance, and then there is ATPase, which pumps out the hydrogen H plus ions. Then there are some chaperones. Chaperones are heat shock proteins that assist in protein folding. Then there are bacteria which internally produce certain acidic or basic metabolic waste. Matlab, in case of acidophiles, they will produce certain basic metabolic waste which will help them to maintain or balance out the acidic effect. Then, in case of alkalophiles, they will produce acidic metabolic waste to balance out the basic effect. And like bacteria, they show buffering effect. Like they contain buffers such as phosphates. Then sometimes amino acids act as a buffer. Peptides also act as a buffer. Next is about barophiles or piezophils. These are the organisms that survive at a very high pressure and like a high hydrostatic pressure. They are also called as barotolerant. Barotolerant example is Moritella cyanosia. So this is an example of barotolerant organism. Now, if another, like, these are barotolerant, the barophiles can be barotolerant or pisophiles. So other organisms which come under barophiles include Photobacterium, Salmonella, and amphipods. Then are certain organisms that are called as thermobarophiles. Thermobarophiles, they will survive at a high temperature, and barophiles, they will survive at a higher pressure. Examples of this include Pyrococcus species, Methanococcus species, etc. So last classification was based on pressure. Now let us see a classification based on the concentration of salt, that is NaCl. And these organisms are called as halophiles. Halophile matlab high salt concentration in a bacteria. Halophiles can be further classified as non-halophiles. Non-halophiles example is E. coli. Then there is halotolerant, which can tolerate some amount of salt. And here it is Staphylococcus aureus. Then there is halophiles, which is Vibrio fischeri, and extreme halophiles, which live under only high salt concentrations, is Helicobacter, Helicobacter, Selenium, Helicobacter Selenium, ऐसा ही है उसका कुछ. Helicobacterium Selenium, हाँ, Helicobacterium Selenarium. This is also H. pylori is also one of them. Next is about xerophiles. Small point about xerophiles. Xerophiles are the bacteria which grow at low water concentration, that is dry environment. Then, in case of osmotic pressure like hypertonic and hypertonic solutions. Hypertonic में क्या होता है? Osmotic pressure is high, and hypotonic में osmotic pressure is low. Sorry, hypo पहले लिखा है, so hypo is low and hypertonic is high. Hypertonic and hypotonic. So hypertonic में solute concentration inside is low. So inside solute concentration is low. So क्या होगा कि बाहर का solute अंदर आ जाएगा, water बाहर जाएगा. So it will lead to exoosmosis. As a result, water goes out. So cell क्या होगा? Cell will shrink and cause plasmolysis. क्यों? क्योंकि water moves out. And in case of hypotonic, the solute concentration outside, inside is high. So water will move inside. So cell क्या होगा? It will swell. Swell होके क्या हो जाएगा? It will finally burst out and क्या हो रहा है? Water is inside. So endoosmosis हो रहा है. Then there is isotonic also, wherein the osmotic pressure of inside and outside the is same, the solute concentration is same, and there is no osmotic pressure difference. So depending upon the osmotic pressure, it is called as osmotolerant. When there is high, which can tolerate high osmotic difference. So they grow over a wide range of H2O concentration or osmotic pressure. Example of such bacteria is Aeromonas and Staphylococcus species. Now depending upon the सेल वॉल को हम लोग इन डिटेल्स एवरी डिटेल ऑफ द सेल वॉल हम लोग पढ़ेंगे इन द सेल बायोलॉजी प्रोकैरियोटिक सेल बायोलॉजी अंडर माइक्रोबायोलॉजी इन द नेक्स्ट लेक्चर्स. बट अभी फिलहाल जस्ट क्लासिफिकेशन के लिए क्लासिफाई कर रहे हैं हम लोग ग्राम पॉजिटिव एंड ग्राम नेगेटिव सेल वॉल में. इट इज गिवन बाय सर हैंसन ग्राम. तो यहाँ पे ग्राम पॉजिटिव और ग्राम नेगेटिव में डिफरेंस क्या है? It is that here that is a very large area for peptidoglycan. But in case of gram negative, it contains peptidoglycan as well as some lipopolysaccharides. So these are lipopolysaccharides that are present, which are absent in gram positive bacteria. Also, lipotecoic acid is present in gram positive bacteria, and teichoic acid is present in gram positive bacteria. Then in gram negative bacteria you can see a periplasmic space that is absent in case of gram positive bacteria. Also point to be noted is that lipoteichoic acid, it is mostly attached or associated with the phospholipids. यहाँ से अंदर जाके यहाँ पे आ रहा है phospholipids. So it is mostly associated with the phospholipids of the plasma membrane along with the peptidoglycan. Peptidoglycan के साथ साथ phospholipid को भी अटैच कर रहा है. लेकिन टिकोइक एसिड देखो, टिकोइक एसिड सिर्फ पेप्टिडोग्लाइकन लेयर तक ही है. इट इज स्टिल यहाँ यहीं तक आ रहा है. टिकोइक एसिड थोड़ा लाइट कलर में और लिपोटिकोइक एसिड इट इज प्रेजेंट इन अ डार्क पर्पल कलर. सो टिकोइक एसिड goes deep into phospholipid and peptidoglycan layer. And here also, like, you can see two membranes over here, two phospholipid membrane in case of gram negative bacteria. These are the major differences in gram positive and gram negative cell wall. Both lipotecoic acid and teichoic acid were joined via मतलब linked via a covalent bond to the respective phospholipids and the peptidoglycan. Teichoic acid to the phospholipids and lipoteichoic to both peptidoglycan layer and the phospholipids. Teichoic acid is like it contains molecule of glycerol and it has a negative charge. So this was about the cell wall. So I think we should end with this lecture. अभी तक क्लासिफिकेशन खत्म नहीं हुआ, जस्ट थोड़ा सा बाकी है और थोड़े क्वेश्चंस भी कर लेंगे नेक्स्ट लेक्चर में. सो सी यू इन द नेक्स्ट लेक्चर. So today let us start with the classification, continue with the classification. So अभी classification जो है आखिरी है, this thing it is based on the flagella. It is the number of flagella that is present. So ये भी याद रखना है कि बैक्टीरिया फ्लैजेला का साइज क्या है. इट इज 10 to 20 माइक्रोमीटर्स लॉन्गर एंड 0.01 to 0.02 माइक्रोमीटर इन डायमीटर. इससे भी क्वेश्चंस आए हैं. Then there is bacterial flagella is of two types, plural flagella and lateral flagella. Polar flagella is the one that is important. It is responsible for motility, then sensor of the and sense as a for the contact with the surface. ये सब करता है polar flagella. And lateral flagella is an alternative flagella, मतलब present है या ना हो, मतलब कभी कभी होता है. And condition inhibitory rotation of the polar flagella. Then classification, agar it is the bacteria does not have a flagella, it is called as atrichous. And the example over here is cocci. If it has only one monoflagella, it is called as monotrichous. Example is Pseudomonas, Theobacilli or Vibrio cholera. Then there is like एक side में अगर more than one flagella, it is called as Lophotrichus. Example Pseudomonas, Spirilla. And if there is one flagella on each side, it is Amphitrichus. And the example is Nitrosomonas. This is an important example. Then Petritrichus is the one wherein the flagella is present on all the surfaces of the bacteria. Example E. coli, Salmonella typhi. Then there is Cephalotrichus. Cephalotrichus is also called as Amphilophotrichus. So इसका example खुद ढूंढ लेना. Another important concept is surface to volume ratio. Surface to volume ratio of a bacteria. मतलब बैक्टीरिया के surface, entire surface to its volume, उसका ratio होगा हमारा surface to volume. ये एक formula है, मतलब surface area of a bacteria to the volume of the bacteria. The surface area, bacteria क्या होता है? Usually a circular as a sphere. The sphere का surface area as formula is 4 pi r square. Or volume of a sphere is 4 by 3 pi r cube. So ये क्या है? Cancel हो जाएगा. What is left is 3 by r. So surface volume ratio of a bacteria is given by this formula. Now, smaller cells will have higher surface to volume ratio and like higher surface to volume ratio, meaning smaller cell, and like the growth is faster. This is एक year question आया था, one numerical. Okay, let us solve some previous year questions related to this topic now. ये अभी है 2024 का क्वेश्चन, इसी साल का क्वेश्चन है फ्रॉम लाइफ साइंस पेपर. Which of the following use inorganic compounds as an energy source? Heterotrophs. Heterotrophs mostly use organic source of energy. Chemolithotrophs, which use chemical energy source and inorganic source of energy. Chemoorganotrophs, नाम में ही organic आ रहा है. Photoheterotrophs, again organic. So the correct option for this question will be D. Chemolithotrophs. Next question is also from last year's question paper. Which of the following represents abundance of organisms found in the soil? So soil में कौन से organisms mostly found होते हैं? Mostly present will be aerobic bacteria, fungi and anaerobic bacteria. So इसका option होगा option B. I think this is also a question of 2024. So as you can see, at most like 3 questions have come from this topic. This question में which of the following cocci is or are examples of division in one plane? So one plane में divide होके बस 2 equal half में divide करना है. So उसको बोलते हैं division in one plane. So Staphylococcus, the bacteria are arranged in groups. कोई symmetry नहीं है. In Streptococcus, it is arranged in the form of straight chain. Micrococci, here the bacterial cells are very small and they are arranged in the form of branches. Diplococci is 2 bacterial cells. So Staphylococcus में you cannot divide the bacteria into one plane. But we in Streptococcus we can divide it into one plane. Micrococcus we cannot divide, but Diplococcus we can divide. So it is this question is an MSQ, and the correct options will be option B and option D. Next question is from GATE 2023. It is related to bacterial flagella. Although we will study flagella in details when we study the full structure of bacterial cell, but here which of the following statements about a bacterial flagella is correct? One of the statements. So this is a MCQ. Bacterial flagella varies in the length ranges from 0.5 to 2 micrometers. Flagella are adjacent fibrils with regular patterns, and flagella has in conjugation, and flagella originates from basal body. Yes, flagella originates from the basal body. And conjugation में तो help करने वाला पिल्लाई है. It is not flagella. It is पिल्लाई. And bacterial flagella का length 10 to I think 0.1 to 10 कुछ था. Moving on with the next question. Which of the following genus are spirochetes? So it is a MSQ for 2 marks. Lactospira is a spirochete. ये एग्जाम्पल में लिखा था. Spirulina, it is not a bacteria, it is a food supplement. Spirulina. Then Treponema palladium, ये भी एक spirochete है. ये भी एग्जाम्पल में था. बस कंफ्यूजन बोरेलिया में था और स्पिरुलिना में था. But Borrelia also belongs to the phylum Spirochetes. So the answer for this question is A, B, and D. Next question: which of the following method is used for identification of microorganisms? So we had already learned 16S RNA sequencing is used. It comes under metagenomics and divides bacteria and classifies bacteria. So answer is 16S RNA sequencing. Okay, next question is related to gram-negative bacteria. Which of the following are present in gram-negative bacteria? Mucopolysaccharide, yes, they are present. Teichoic acid, no. Teichoic acid is not present in gram-positive bacteria. It is a specific feature of gram-positive bacteria. Periplasm, yes, periplasmic space is present in gram-negative bacteria. Endotoxin, yes, it is present in the cell wall of gram-negative bacteria. Okay, this is a question from 2022. It is a match the following, which will come for 2 marks. So यहाँ पे बैक्टीरिया दिया है और उनकी मॉर्फोलॉजी दी है और सेल डिवीजन दिया है. मतलब मॉर्फोलॉजी को देख के हम लोग को पता लगाना है कि सेल डिवीजन बैक्टीरिया कैसे करेगा. स्ट्रेप्टोमाइसिस स्पीशीज, हाउ आर स्ट्रेप्टोमाइसिस स्पीशीज अरेंज्ड मोस्टली? So questions like this we can try solving by what we know. Like Streptomyces species, the cells are arranged in the form of chain. So when they will divide, they will form a cell like this. Then one more cell will be formed like this. So finally they will form a chain. One cell, two cell, they will divide like this. So the most probable answer for this option will be option 2 for Streptomyces. Like probability देखनी पड़ेगी. फिर in case of Bacillus subtilis, a simple bacteria which will divide by a normal cell division. Cell will cut into half and two cell will be formed and then they will separate and give two cells. So this, from this I think this will be the answer for this. Bacillus subtilis ये हो ही नहीं सकता क्योंकि shape of the bacillus is like this and this I think this is for Norcardia हो सकता है, ये नहीं हो सकता. Norcardia species का हम लोग को नहीं पता. So we are like sure कि फर्स्ट वन का R हो सकता है एंड सेकेंड वन का T हो सकता है. So is there an option over here? T second one and R is first one. T is second one, R is third one. R is first one, T fourth one. So T is first one. So answer will be answer option A. Next question. Which of the following are used as an electron donor for carbon dioxide reduction during photosynthesis in purple sulfur bacteria? Purple sulfur bacteria, यहाँ पे जो इलेक्ट्रॉन डोनर मोस्टली होता है वो होता है इनऑर्गेनिक हाइड्रोजन सल्फाइड हो सकता है, थायोसल्फेट हो सकता है या फिर सल्फाइड हो सकता है. मीथेन नहीं हो सकता, ऑर्गेनिक नहीं हो सकता. Okay, so let us solve this numerical. This is the same numerical that we had studied the formula for surface to volume ratio. So consider a spherical bacterial cell with diameter is 2 micrometer and a eukaryotic cell of spherical shape with a diameter of 20 micrometer. So एक bacterial cell है और एक eukaryotic cell है. The surface area to volume ratio of the bacterial cell is dash times more than the eukaryotic cell. So surface to volume ratio का formula क्या था? Surface to volume ratio was 3 by R. So 3 diameter divided by 2. So it will be diameter 2 by 2. And इसका कितना होगा? Surface to volume ratio, it will be 3 divided by 20 by 2. So this will be 3 and this will be 0.3. So the surface volume ratio of the bacterial cell is dash times more than that of the eukaryotic cell. So how many times more? It is 3 by 0.3, which is 10. So answer is 10 times more than the eukaryotic cell. So I think we have done almost most of the questions till for the previous year questions. So let us stop for today. And topic, we have not finished Archaea as yet. So we will complete Archaea and its classification and complete Archaea in the next lecture. Numerical, I guess.